Hello guys, welcome to this YouTube channel. A good day to everyone watching this video. In this video, I am going to discuss the answer key for the GATE Excel Biochemistry section of the 2017 paper. So let's get started. So the first question is, the molecular weight of a protein as determined by native page is 400 kilodalton. This protein, when run on a non-reducing STS page, gave a band of 200 kilodalton, and on a reducing STS page gave a band of 100 kilodalton. So in case of a non-reducing condition, what do you mean by non-reducing is that the disulfide bonds are intact. Disulfide bonds are intact, and here you get a band of 200 kilodalton, which means basically the 400 is broken down into 200 and 200. And in case of a reducing STS page condition where the disulfide bonds are reduced, so in case of a reduced STS page, you have a band of 100 kilodalton. So what does this mean? This means that this protein has 4 subunits. So how? 400 divided by 100, which is equal to 4 subunits, and 2 sets are linked by 2 disulfide bridges. So if I have 4 subunits like this, hypothetically, let us say, if this is the case, then let's take this as one set and this as one set, and there is disulfide bonds between this. So in case of a non-reducing page, you have only only the cleavage like this, and as a result you have this 200 kilodalton bands. However, when you have a reducing STS page condition, even this is also cleaved, and hence you have 4 into 100 kilodalton, and that ultimately gives you one thick 100 kilodalton band. So that's the reason why the correct answer here is option A, that is 4 subunits, of which 2 sets are linked by 2 disulfide bridges. I hope this is pretty easy to understand. Hope you don't get confused. Moving on to the next question. Which one of the following techniques cannot be used to determine the sequence of a novel protein? So first of all, de novo sequencing by ESI-MS, yes, this can be used because it already says de novo sequencing in the name, and ESI-MS is a mass spectrometry. Yes, it can be used. Edman degradation, of course, this can also be used in determining the terminal ends of the protein. Sanzo sequencing, which is like the conventional approach of deducing the sequence of a protein. Yes, that is also used. However, peptide mass fingerprinting cannot be used to determine the sequence of a novel protein. So the correct answer here is peptide mass fingerprinting. So this you can, for this, if you don't know what the principle of peptide mass fingerprinting is, you can definitely, guess the answer or guess the correct answer for this question by deducing method. Moving on to the next question. Which type of polyacrylamide gel can be used for analyzing the 4 different proteins listed below? So here you have 4 proteins named PQRS, and if you can see, the molecular weight is also given and the isoelectric points are also given. So it's going to be a gel with isoelectric focusing basically. So here if you look at this, the isoelectric point varies from 4 to 8. So 4 is the least one and 8 is the highest one. So of course you cannot have the gel which has a pH of 4 to 7, because then we cannot get the band dissolved for 7.5 and 8. However, the answer has to be the pH in the range of 3 to 10. That is correct. However, you also need to decide which percentage of gel would be ideal for this. And since it's a high molecular weight protein of 60 kilodalton and 45 kilodalton, then this is the right one, that is 12% gel, because 20% gel, the pores will be too small for these proteins to migrate, and hence a 12% gel will be more ideal for proteins of these molecular weight, and the pH 3 to 10 ideally covers the range between 4 to 8. So the correct answer here is option C. Moving on to the next question. So here, let me just zoom in. The number of fragments generated when the peptide, so this is the big peptide sequence, is subjected to complete digestion with trypsin R. So when you have a complete digestion with trypsin, you should know where the cleavage sites for trypsin are. So trypsin basically cleaves the peptide bonds at the C-terminal site of lysine and arginine. So these are the one-letter amino acid codes for lysine and arginine respectively. lysine and arginine residues. However, it doesn't cleave when this lysine or arginine is bonded to proline. Why? Because proline is an amino acid and it offers steric hindrance. It has a very rigid structure. So due to steric reasons, trypsin cannot cleave when this lysine or arginine is immediately followed by proline. So here the sequence of the peptide is given. Since they haven't mentioned anything, it is safe to assume that this is from N-terminal to C-terminal as the conventional approach. So here let's look for lysine and arginine residues. So let's just circle them. Here you have one lysine, here you have another lysine, here you have an arginine, and here you have another arginine. So here this lysine is immediately followed by proline. So here trypsin cannot cleave. And similarly here the arginine is followed immediately by proline. So again here trypsin cannot cleave. So the trypsin basically cleaves here and it also cleaves here, leaving one band, one peptide. This is going to be the second peptide and the last is going to be the third peptide. So here the total number of fragments, the peptide fragments generated when this big peptide is subjected to complete digestion with trypsin are 3. So this is a very simple question if you know this particular fact, and of course this is a very important thing, otherwise it will be easily misleading for you to even include these sites of digestion. Moving on to the next question. Puromycin is a structural analog of, the correct option here is answer B. I'm not going into the mechanism of how puromycin inhibits translation. However, it is important to know that it is a structural analog of tyrosyl tRNA and it inhibits translation. And the mechanism of action of different antibiotics is something that is frequently tested in most of the biology competitive exams. Just to keep this video short, I'm not going into the mechanism. However, the correct answer here is option B, that is tyrosyl tRNA. Moving on to the next question. Which one of the following enzymes is responsible for arsenic toxicity? So here the correct option is pyruvate dehydrogenase. So this enzyme is important for the conversion of the pyruvate into acetyl-CoA for entry into the citric acid cycle, and this is a major target for arsenic toxicity. So the correct answer here is pyruvate dehydrogenase. Moving on to the next question. Which one is true for the Calvin cycle? So again, just to keep this video short, I'm not drawing the entire Calvin cycle here. However, it is important to know the different steps in Calvin cycle and the associated facts related to it. And here the correct option is, this is a reductive pentose phosphate cycle. So this one is true for Calvin cycle. However, if you look at the other options, glycerol 3-phosphate is generated, which is not true. CO2 is not consumed in the cycle, which is again not true. And even ribose 5-phosphate is a carboxylation substrate in the cycle, that is also not true. So if you look at the Calvin cycle, you will know that this is a reductive pentose phosphate cycle, and that is true for this particular question. Moving on to the next question. Administration of primaquine causes severe hemolytic anemia because the mechanism of action of this primaquine is to increase the demand for NADPH to such a level that the cells can't meet. So basically this NADPH is a metabolite that is also involved in the energy pathways, and this primaquine basically it increases this demand for NADPH to very high levels that the cells can't meet this demand, and the cells and these RBCs, that is hemolytic anemia, results in this disruption of all these erythrocytes. So that is how the drug acts. So it is not through inactivating the glutathione peroxidase, or it doesn't reduce glutathione levels of erythrocytes. Instead, it increases the NADPH levels to such a high extent that the cells can't meet this demand. Moving on to the next question. Which one of the following will not form a lipid bilayer? This is a very simple question. If you know the basic lipid composition of the upper and the lower of the cell membrane, basically layers of the cell membrane. So you have cholesterol that can form a lipid bilayer. Of course, phosphatidylethanolamine can also form, and even phosphatidylserine can also form. Triacylglycerol cannot form. So this is cannot form a lipid bilayer. So the correct option here is triacylglycerol. Moving on to the next question. Yes, which one of the following features is not appropriate for the Fab fragment of IgG? So first of all, you should know how this Fab fragment is generated in the antibody, which includes the antigen binding site. So that is true. It also contains an intact tail chain. Yes, that is also true. And two Fab fragments are formed from one IgG molecule. Yes, that is also true since you have two variable regions here, and it mediates complement fixation in the intact IgG molecule. This is not true. So the correct option here is option B. Moving on to the next question. Here, so the duration of the DNA synthesis, that is S phase, in plant cells is 11 hours, and the DNA is replicated at a rate of 100 base pairs per second per fork. A plant species has about 3 × 10 power 10 base pairs of DNA per genome. The number of bidirectional forks per genome required for replication will be what? So first of all, you need to be very clear with how you do this question. So first of all, what is the length of the genome? It is 3 × 10 power 10 base pairs. So that is given. That is equal to length of the genome. Yes. And now, one fork, if you take, it can replicate 100 base pairs per second. So basically, the S phase, if you look for this particular plant cell, it is for 11 hours. So 1 hour has 3600 seconds. So this is a basic fact. So 11 hours will have 11 × 3600 seconds. Okay. So for this many seconds, what is the total number of base pairs that one fork can replicate? So for one fork, for the entire S phase, what is the number of base pairs it can replicate? So that will be 100 × 11 × 3600, right? So totally you have these many number of seconds, and for 1 second, one fork can replicate 100 base pairs. So for the entire S phase, one fork will replicate 100 × 11 × 3600 base pairs. Okay, excellent. Now we need to know the number of forks. So how will you know the number of forks? So you have the total length of the DNA of the genome is 3 × 10 power 10. So if you divide 3 × 10 power 10 divided by the number of base pairs one fork can replicate for the entire S phase, you will get the number of forks. So that will be 3 × 10 power 10 divided by 3600 × 11 × 100. So if you calculate this, you will get 10 power 6 divided by 132. The answer roughly comes to 7576 forks. So you basically need 7576 bidirectional replication forks for the complete replication of the genome for the entire S phase. So this is a very simple question. If you don't get confused between what to divide and how to divide. Yeah. Moving on to the next question. In a PCR reaction with one double-stranded DNA of 600 base pairs, the nanogram of DNA produced after 40 cycles of amplification will be. So here the calculation can get a bit messy, however let's solve. So for a PCR reaction, the formula is N, which is equal to the number of DNA molecules that is produced after so many number of cycles, is equal to N0, which is the initial number of DNA molecules that you add for the reaction, into 2 power n, where this n is the number of cycles, which is 40 here, right? Okay, so n equal to 1, since we start with only one double-stranded DNA. So 1 × 2 power 40. So this many number of DNA molecules are produced, right? Okay, so one DNA molecule has 600 base pairs. So 1 × 2 power 40 DNA molecules will have how many base pairs? That will be, so you will write total number of base pairs will be equal to 1 × 2 power 40 × 600, right? Excellent. So now you should know the molecular weight of one base pair. So one base pair weighs 660 Dalton. 660 Dalton or 660 gram per mole. So this is the weight of one base pair. However, we have these many base pairs. So how do you now figure out the weight? So that will be equal to, weight of the DNA molecule that is produced after 40 cycles will be equal to 1 × 2 power 40 × 600 × 660 gram per mole. So 1 mole is equal to Avogadro number, which is 6.022 × 10 power 23. Now, if you look at this calculation, this can get really messy because as you see here, you have a 2 power 40 where it's a very big number and you have a denominator with this. So, let me just write this weight as say x. So, now I'm going to take logarithm on both sides. So, log x will be equal to log of this entire number, right? So, that will be equal to 40 into log 2. So, if you know the formula for logarithms, that is log of a power b equal to b log a. So that's what? So log 1 is 0. So log of 2 power 40 is 40 into log 2. Plus if you have log ab, it is equal to log a plus log b. So that's why when there is into, I'm just adding the logarithm. I'm just taking the log inside and then adding all the terms. So that will be log 600 plus log 660 minus, if you have a division, if it is a by b, it is log a minus log b. So you will now subtract. So log of 6.022 minus 23 into log 10. Log 10 is 1 because the base is 10 and hence it's going to be 1. So here you will have 40 into log 2 plus log 600 plus log 660 minus log 6.022 minus 23. Because log 10 is 1. So if you calculate all this, you will get the answer to be minus 6.14. Okay. However, now you need to get x, which is the weight. So x is equal to 10 power minus 6.14 grams. And they want the answer in nanograms. So how would you convert this? So it will be 10 power minus 6.14 into 10 power 9 nanograms. Because 1 gram is equal to 10 power 9 nanograms. This we know. So here if you subtract, you will get 10 power minus 6.14 plus 9, that is basic exponential, that will be equal to 10 power 2.86 nanograms. So if you now use the calculator on how to calculate 10 power 2.86, it will give you an answer to be 724 nanograms. So this is the correct answer for the following question. So here the calculation can get a bit messy even though you know how to do this question. So if you know basic logarithms and then take log and anti-log on both sides, you're going to get the answer easily. Anyways, moving on to the next question. So here a solution containing GTP has molar extension coefficient of 1.55 into 10 power 4 mole inverse decimeter cube centimeter inverse at a given wavelength. The concentration of GTP solution is 1.29 into 10 power minus 5 mole decimeter cube inverse. Yes. The absorbance of GTP solution in 1 centimeter cubed at the same wavelength will be. So here again we use Beer-Lambert's law, which is frequently tested. So please remember this law. It's going to be an easy cakewalk. So A is equal to epsilon into C into L, where A is the absorbance, epsilon is the molar extension coefficient, C is the concentration, and L is the path length. So you just have to substitute because here all the units are in the correct order, so they are all matching, so you don't have to do any conversion. That will be 1.55 into 10 power 4 into 1.29 into 10 power minus 5 into 1, because it's a 1 centimeter cubed length, L is 1. So that will give you an answer approximately 0.2. So here the absorbance will be 0.2. Very simple question. Moving on to the next question. Which one of the following is not true for class 1 MHC protein? So MHC class 1 protein are polymorphic. Yes, that is true. T cell receptors recognize MHC class 1 proteins. Of course they do. MHC class 1, which particular T cell receptors? Yes, which particular T cells? It's the CD8 receptors which actually recognize MHC class 1. Next, MHC class 1 protein are displayed on the surfaces of nucleated vertebrate cells. That is also true. However, what is not true is the beta-2 microglobulin is covalently associated with MHC class 1 protein. That is not true. So if you look at the structure, that is not covalently attached. So the correct answer here is option D, which is not the case for MHC class 1 protein. Moving on to the next question. In an enzyme catalyzed reaction, the initial reaction velocity is only 1/4 of its maximum velocity. If the substrate concentration is 3 into 10 power minus 3 millimolar, the value of Km in micromolar will be what? So this is a Michaelis-Menten's kinetic equation, that is V equal to V max into substrate concentration divided by Km plus S. So here they have given that this V, which is the initial reaction velocity, is 1/4 of the maximum, that is V max. So substituting V equal to V max by 4 will be equal to V max into S divided by Km plus S. Cancelling V max on both sides, which is mathematically possible. So here you get Km plus S equal to 4 times substrate concentration. So Km will be equal to 3 times substrate concentration, which is 3 into 10 power minus 3 millimolar, which is 9 into 10 power minus 3 millimolar, which is 9 micromolar, because 1 millimolar is 10 power 3 micromolar. So here you have the correct answer to be 9, because they have asked the answer in micromolar. Very simple. Moving on to the next question. So here, match the following enzymes in column 1 with their cofactors in column 2. So here, the correct answer here is option B, which is this. So here, if you look at the option, pyruvate decarboxylase has thiamine pyrophosphate, TPP. If you know the way how pyruvate decarboxylase works, that is the cofactor for this. Glyceraldehyde 3-phosphate dehydrogenase, that is Q, has NAD plus as the cofactor. If you remember, NAD plus gets converted to NADH. And pyruvate carboxylase, which is R, has biocytin as the cofactor. Yes, that is also true. And the glucose 6-phosphate dehydrogenase has NADP plus as the cofactor. So the correct option here is option B. So if you read these glycolysis and TCA cycle and even gluconeogenesis and all these pathways, it is important that you also remember the cofactors, because these are some of the questions that I frequently see in especially the GATE Excel biochemistry papers, even when I was attempting my exams. Moving on to the next question. So match the molecule in column 1 with its function in column 2. So here the correct option is option A. So here cholera toxin, it basically modifies the stimulatory G alpha in the G protein, GPCR, and the pertussis toxin, which is Q, modifies the inhibitory G alpha. Yes. IP3, it is mainly involved in the calcium level, so it increases the intracellular calcium levels by increasing the secretion from the ER. And then the caffeine, it inhibits the CAMP phosphodiesterase. So that's and it also interferes with the cell cycle progression. So the correct answer here is option A. Moving on to the next question. So here, in an in vitro dehydrogenation reaction of succinate catalyzed by succinate dehydrogenase, malonate is added. Which one of the following curves represent the effect of malonate on the catalysis of succinate dehydrogenase? So it's basically a competitive inhibition reaction, and the correct answer here is option A. So here, if you look at this graph, so here this particular graph will represent that malonate is inhibiting this particular reaction of succinate being converted to the next product by the succinate dehydrogenase reaction. So the correct answer here that describes this inhibition reaction is the option A. Moving on to the next question. Cardiotonic steroids have the ability to strengthen heart muscle contraction due to the fact that these steroids, so how do these steroids work is they basically it basically prevents the dephosphorylation of this Na plus K plus ATPase. So when this happens, the level of Na plus increase inside the cell. So when this happens, the intracellular sodium ion concentration increases, and when this happens, this will slow down the Na plus K plus exchanger. So Na plus Ca2 plus exchanger. So when the intracellular concentration of Na plus increases, this leads to slowing down of Na plus Ca2 plus exchanger. So when this happens, Ca2 plus is going to accumulate. Ca2 plus accumulates inside the cells, and when this happens, contraction is resultant. So as a result, you will have heart muscle contraction. That is how these cardiotonic steroids work. They basically indirectly increase the Ca2 plus levels by interfering with the dephosphorylation of Na plus K plus ATPase, which is K plus dependent. So that's how these cardiotonic steroids work. Moving on to the next question. So here, this is a really good question. A newly isolated circular plasmid gave two bands of 3.2 and 3 kb on digestion with EcoR1 and two bands of 5 kb and 1.2 kb on digestion with BOM-H1. Double digestion with EcoR1 and BOM-H1 yielded 4 bands of 2.6 kb, 2.4 kb, 0.8 kb and 0.4 kb. So digestion with SAL1 leads to disruption of the ampicillin resistance gene cassette. So the correct restriction map is. So here, if you don't read the full question, you're going to waste time with this question. So here, there is one hint that is like really good for this question, that is digestion with SAL1. It leads to the disruption of the ampicillin resistance gene cassette, which means this SAL1 has to be in the ampicillin resistance gene. If you look at this first diagram, SAL1 is not on the ampicillin resistance gene. So this is not the option. And here if you look at again, SAL1 is away from the ampicillin resistance gene, so that is also not the correct answer. And similarly here. So this leaves with the correct option, that is option B. So you basically don't even have to solve for this EcoR1 and BOM-H1. Instead, if you read the question fully and just figure out that, okay, SAL1 is in the ampicillin resistance gene for only this plasmid, then this is the correct answer because the other three don't have this. So the correct answer here is option B. If you just follow this entire question properly, you get the answer in 2 seconds. So that's how you save time. So with this, I am completing the discussion for the answer key for the Biochemistry Excel paper, for the 2017 paper. I hope you found this video interesting, and I also hope that you found this video useful. Wishing you all the very best for your upcoming examinations, and hope your preparation levels are also going in full swing. Thank you, and let's meet in the next video. Bye.